Part 2 of process validation, and in our first video we talked about some of the essential aspects of what is necessary for process validation. But let's move on to some case studies so that you can have a broader understanding of how this may be implemented within your establishment. So, what are the steps you need to do to actually enact a process validation? Well, as we mentioned in our first video, you do need to make sure that your HACCP program is complete, and therefore you have been able to identify the hazard that you are going to control. So, are you controlling bacteria? Which bacteria? Do you know its control mechanisms? What about chemicals? Do you know the trace for that chemical and how it gets into your food? Is it something that's done in your establishment, or is it something that's coming from part of your incoming value chain? Do you know what the source of that contamination is? Is it coming from the environment? Is it from poor hygiene? Is it from cross-contamination? Is it your supplier that's introducing the contamination into your facility in the first place? Do be very acutely aware of where potential sources of contamination are coming from, and be very observant, thinking high and low about what are the probabilities of these things occurring and what is the potential impact of them. Identify your outcome. Now, in many cases, facilities say we have a zero tolerance level for contamination in our product. But in other cases, you may say a very small level of contamination is acceptable, and it really is dependent on the hazard. In some industrial programs, Six Sigma, for example, may have defect levels based off of certain statistics. So Six Sigma, they often say we want defects showing up in less than 1 in a million samples. Now again, in the case of any process, you do have your process-related controls that you can manage within your process. So temperature, pH, water activity, oxygen time, use of preservative ingredients, and identify what their potential role is in managing this hazard. These are things that you can directly control within your process. So, for example, are there industry regulations for your commodity or industry standards that define these processing parameters? So, for example, as we mentioned before, there is pH requirements for canned foods. pH critical limit must be below 4.6 for Clostridium botulinum control. Clostridium botulinum being a soil-borne organism, so on virtually every fruit and vegetable that is grown near the soil, there's a possibility of cross-contamination. Or likewise in fish. It is something that is mucked up into the gills of the fish and becomes embedded in the tissues. And from there, you need to identify time and temperature combinations that are sufficient, that are for elimination of the hazard during the cooking and pasteurizing process. We'll talk more at a later point about how we evaluate time and temperature combinations for food products. So, in certain cases, we need to go and build up that body of evidence. Where do we find this evidence? In certain cases, it is a government regulation. In other cases, we need to go and look at the scientific literature, the technical literature, and other previous studies. In case of many industries, they will have what we call blue ribbon studies or blue ribbon inquiries, where they will identify an industry-wide hazard and they will go and do the scientific research on behalf of the industry to be able to identify appropriate means. Again, we mentioned before, in many cases there are regulations where the government or other industry associations have come in and said, this is our standard, you must adhere to it. Government directives are extremely common in this regard, but we've noted over the past 20 years or so, government has gone and said the evidence and the burden of evidence is transferring more away from a set of critical limits to define your own evidence. Again, there's lots of different sources for standards. Again, in certain cases, these may be coming from a processing authority. As we mentioned before, processing authorities are industry experts, specialists. Many times they're professors who specialize in process validation, and they are experts, often with advanced academic credentials, to be able to do the risk assessment and to do, in many cases, mathematical modeling of the food product to evaluate its potential for outgrowth of bacteria or growth of toxin and so on. In some cases, if you're bringing in a new piece of equipment relative to especially thermal processing, but in other cases, a novel pasteurization methodology, the equipment manufacturer, because they want to be able to sell this, may have already built up a research database showing that running the equipment under certain conditions is going to provide pasteurization or death of various organisms under certain run conditions. And equally too, when you're commissioning new equipment, you may be wanting to collect testing data off of the new processed product coming off of it to ensure that there's not any contamination. In certain instances, if you have been preparing a product in your plant for a period of time and you're changing the level of HACCP programming, or you are changing your export requirements, you may have not been required to have the validation evidence, but you may actually already have accrued what's called in-plant historical data. If you've had to send out anything for testing, if you have a clean record in terms of recalls, you may be able to use this sort of burden of evidence as a positive towards your process validation. Another form that's commonly used is when developing a new product is to use what's called a Holden test. This is where on every lot of product or a routine skip lot approach, you're going to be testing the product for that target organism or that target hazard to ensure that the hazard has not carried through to the process. So every lot of product, a sample of product is sent out for testing and show that the hazard is negative within the product. In certain cases, you're going to use scientific data from lab challenge testing, where you send a product to the lab and it's inoculated and the product is processed or held under the conditions which you anticipate the hazard would be negatively influenced, and see if indeed your product formulation or the processing methodology is going to prevent and mitigate that hazard from moving forward. Scientific data can also come from in trial pilot plants. So there are facilities such as Agriculture Canada Food Research Centre at Guelph. They have a pilot plant where you can go in and fill your food up with bacteria and process the heck out of it to see if the bacteria actually survived. Your own plant is better, but again, evidence is evidence, and the more evidence you have, the better off your validation is going to be. You do need to document it. And so in many cases, organizations or establishments will take a combination of approaches when evaluating how much evidence they have. They want to make sure that the control measure and combination of control measures are consistently being implemented. You want to make sure that you have a whole variety of different opportunities. Make sure that the study is done in a very well and comprehensive manner so that you can analyze it, document it, and have it on file for any inspection requirements or HACCP audits. So how do you prove control then? You do need to make sure, just like under any HACCP program, that you have appropriate monitoring and verification of your control mechanisms. So again, making sure in monitoring that you're going and measuring pH, temperature, water activity, and so on, making sure that those appropriate measures are documented, and that in verification you're making sure that monitoring task is being implemented appropriately and completely. I'm going to jump out here and make video number 3, and it will be another case study thinking about directly evaluating the processing of some aflatoxin in tree nuts. Take care and have fun.